No se oye nada.
Okay, Gustavo is going to solve the problem with his microphone, so I will continue with my presentation. Okay, good morning, everyone. Today I would like to talk about this article called Three-Dimensional Microengineered Vascularized Endometrium on a Chip. The idea of these authors was to create a 3D model that recapitulates the functions of the endometrium. So the endometrium, in the human endometrium, undergoes a cyclical development. So the first point to test was to culture endometrial stromal fibroblasts in different layers. So first I would like to show you the structure of this model. As you can see, there are 5 channels, parallel channels, which are separated by these posts. And in the third channel, you can see that the posts have this special shape, which is called crown shape. And here, the pink channels represent the channel for the medium. And here you see the different layers, the fibrostomal channel, the stroma angiogenic sprout channel, the vascular network channel, and another media channel. Here you can see the development of the vascular network. For example, in day 1, in day 6, the vascular network grows, and in day 8, you see that it passes through the posts, and it starts proliferating into the stroma channel. They have labeled the cells with these markers. So here in green, you can see the actin, which shows us the cytoskeleton, and the epicam, it shows where the epithelial, the endometrial stromal fibroblasts are. In red, you can see here, and they are going to measure the thickness of these layers. And finally, with the CD31 marker, they show the epithelial cells. Okay, so they are going to test different treatments, and they are going to emulate the same conditions of the phases of the female cycle. So here you see the proliferative phase is characterized by a high production of estrogen. Then the secretory phase is characterized by the high production of progesterone. And here you see a schematic of the experiment. So in day number 1, they culture endometrial stromal fibroblasts and endothelial cells. And at day 8, they culture endometrial epithelial cells. Then they start the hormone treatment, and after 12 days, they fix these cells and they perform the immunostaining. So here you see the thickness of these endometrial epithelial cells with this marker, the epicam, and they measure this thickness. So you can see that when the cells are exposed to estrogen, the thickness increases, and it's significantly different from the control and from the other treatment when it's exposed to both hormones, estrogen and progesterone. Okay, and they also characterize the fibroblast acting area. As you can see here, they divide in these two layers, and you can see the fibroblast acting area. It's measured by image analysis. They also characterize the vasculature, the vascular network with the CD31, and it's also measured by the image analysis. So they have achieved to create this model of the endometrium, and then they test this drug called levonorgestrel, which is the most used emergency contraception drug. So what does it? What it does is to sheet this layer of the endometrium. So you can see here that they have applied different concentrations of this drug, and they measure the cells covered, the area covered by dead cells with the propidium iodide. So here in red, you can see the dead cells, and the area of these dead cells increases according to the concentration of this drug. So here you see the schematic of the experiment. So in day 11, they apply this drug, and they measure this blood vessel regression index. It means how the vascular network has decreased its size. So here you can see the index, and it also diminishes as the concentration of the drug increases. So this is a great model to study how these drugs, it's a great model for drug screening for toxicity testing in the field of female reproductive. Okay, thank you very much.
Okay, hi. Good morning, everyone. I'm gonna present my paper. Okay, this is the title, a fully inkjet-printed microfluidics, a solution to low-cost, rapid three-dimensional microfluidics fabrication with numerous electrical and sensing applications. Okay, in this case, the authors use a different technique to fabricate different prototypes of microfluidic devices. The idea is use different substrates. We will see in the next pages what is the substrates, but the most used is glass, and then they use ink. That means that they print this configuration with SU8 ink, and then they printed the channels with PMMI ink that is homemade by the authors. They use different layers in order to build a microfluidic device in 3D. The idea of this paper is print different layers or characterize different layers from ES8 ink and PME ink. Okay, we'll see. Okay, they use different sizes of the channel in order to characterize what is the difference between these numbers, because they increase 5 microns in order to print the microfluidic channel. The microfluidic channel, remember, it was constructed, it was printed by PMMI ink, and they print 15 layers, and then they measure the height of this, and they almost found a linear relation with in the figure number 3 in part C. You will you see the this linear behavior. But what happened with the shape of this division channel? If we return to this part, you will see this is the cross section of this microfluidic. In this case, they use LCP substrate. This is SUOH ink, and this is the channel. If you see, it has this behavior, and they found what is the explanation of this. Because if you see the perfil between height and weight, you see this behavior, and they say that the problem is it's not a problem, but this is the behavior of the ink when you use heat, because when you have a drop of ink at the borders, the evaporation is higher than the center. It means that the flow is in this way. For this reason, at this part, the evaporation is high, and the shape of the channel is like this. No, they try to simulate this behavior in the figure in the part, but the real measures are in the 12 layers, also in ESUH ink, and they found that in this case the layers of the SU8 it was 12 layers, and they measure also the height of this, and they found also this behavior. For this reason, they have a few limitations in order to fabricate this microfluidic. But the good thing of this kind of microfluidic fabrication, it's like you can insert electrodes. You can isolate, or you have an isolate level that is indispensable when you want to introduce electrodes between the layers in order to measure different things. For this reason, this is the model that they use for measure the electricity or some another things. They build a 3D microfluidic device, but they have the ability to measure with electrodes, and it's quite easy. They perform different prototypes, as I told you. This is the different substrates that they use. The first is PET, and then the PAT, Kapton, glass, LC Cooper, PET and again, and LCP Cooper. They perform these kind of prototypes, and some of them use electrodes. Yeah? And this is the configuration of the one of these. They use in this case copper like as a substrate, and they measure the capacitance between when the channel has liquid or without liquid, and they have these lines with the impedance with this. One of these is the simulation. The red line, the green line is the simulation, and the blue and the red is the measure with liquids. If we see in G part, we have this is also the simulation, the blue line, and you use different liquids in order to measure the frequency of this, and this is the way to characterize also liquids inside of the microfluidic, because the perfil that you see is different between different fluids. You know? Also, you can also measure the growth of the cells. Also, it's possible to measure the dead cells. There are different applications when you introduce electrodes between the channels. Okay, thank you for your attention. This is all.
Okay, this is my paper. microfabricated array of clamps for immobilizing and imaging C. elegans. This article describes the manufacture of a microfluidic device to immobilize worms. The chip consists of a matrix of 120 microchannels with immobilized the worms. 150 worms are immobilized in 15 minutes. The process was sent harmful to the worms. The worms reproduce normally. This part, C. elegans is shaped like an elongated cylinder, measures approximately 1 per 2 millimeter long and 50 millimeter wide. Um, Worms were captured between the microchannels, and the pressure difference between the inlet and outlet of the device was used to write the liquid flow. Here is a diagram of the pressure distribution along the microchannel. Um, in this part, channel will range from 100 millimeter to 10 millimeter length of, of 5 millimeter. Channels were designed to accommodate worms of different size. Shows the design of a matrix of 4 clamps in each bifurcation. The path of the worm is determined for the flow radio. And in order to analyze the 4-channel flow distribution, it is necessary to follow the distribution of the liquid from the inlet to the outlet. Okay. By increasing the bifurcation from 2 per 7, 128 clamps were designed. The height of the channel is 55 millimeters. The holes were 1 per 5 millimeter. Show an illustration of the experimental setup. The pressure difference on the chip was raised by connecting the output to a vacuum source or through a polyethylene pipette. The inlet was connected with a Pasteur pipette, then the M9 buffer and the worm suspension were loaded, and this part, 116 worms are immobilized between the bone. More than 90% of the clamps containing an immobilized worm. 11 clamps don't contain worms. This is due to a decrease in the input flow on the chip. Not all reach the same position along the clamps, and the conclusion, the series of clamps immobilize 100 worms at a time, and thanks to the chip, the morphology of the worm can be analyzed, as well as identify natural variation in physiology. Thanks.
You can see my screen.
Yes, we see it.
Thank you. Okay, good morning, everyone. I will talking about of this article called Glucose-induced abnormal echolaging rate in C. elegans. Okay, Glucose in chronic excess leads to the dysfunction of multiple organ systems, a phenomenon termed glucose toxicity that underlies the development of diabetes and pre-diabetes. The elegant lifespan assays is a good model system to evaluate glucose toxicity and develop more efficient therapies for diabetes. Here, they document another phenotype associated with high glucose feeding, a decreased egg laying rate that can be performed more easily than the lifespan assay, and therefore might be applicable for large-scale drug screening against glucose toxicity. They use worms on final larval stage L4 were placed on NGM containing the glucose at the indicated concentrations of 0 and 100 millimolar, and they notice a reduced number of egg release in the presence of high concentrations of the glucose. Compared with that in the absence of glucose, uh, although glucose had no effect on the onset of egg laying. They therefore studied egg-laying behavior using three assays protocols. First, they evaluated the egg-laying rate of individual animals. In the second assay, they scored the number of egg layings in utero, found that the number of the unlaid eggs in glucose-fed animals was higher than that of control animals, suggesting that high-dose glucose did not affect oogenesis but reduced the speed of egg laying. And in the third assay, they measured brood size. 100 millimolar glucose-fed worms showed no significant difference in brood size compared with controls. Taken together, the findings show in this figure that feeding 100 millimolar glucose affect egg laying behavior without altering the rate of gametogenesis and fertilizations. Egg laying is controlled by the hermaphrodites' specific neurons. Cholinergic signaling in the HSN suppresses expressions of the essential serotonin 5-HT biosynthetic gene TFH1, thus inhibiting egg laying. And using this 5-HT deficient mutant exhibited egg laying defect or EGL phenotype. In this figure, you can see that the glucose feeding had no further effect on the egg laying rate of these mutant worms. They're suggesting that the glucose-induced EGL phenotype of N2 worms was dependent on serotonergic signaling. Further, they use SER1 worms. SER1 is a receptor that plays the clearest role in egg laying behavior to examine the effect of glucose in egg laying, but did not show significant difference between the basal rates of egg laying in the wild type and mutant. You can see in this figure the glucose could affect the upstream serotonergic neurotransmission that lead to the activations of the vulval muscle function. And 5-HT dose dependently increase the egg laying rate even in the presence of glucose and completely contracted the effect of glucose at concentrations of 5 millimolars. These results suggested that high glucose did not impair the vulval muscle function, that consequently influence egg laying. They tested the possibility of well-known anti-diabetic drugs called counteract the effect of glucose use two different classes of oral anti-diabetic drugs, buformin and pioglitazone. It has been shown using C. elegans and high glucose increases uh, reactive oxygen species that allowed accumulations of molecular damage and contribute the numerous human diseases, including diabetes. In this figure, you can see the glucose lowering action of buformin and pioglitazone may decrease the formations of ROS, thereby ameliorating egg laying. In conclusions, the present results anticipate that this simple and rapid in vivo assay system could be potentially useful for screening of novel anti-diabetic drugs candidates. And that is all. Thank you.
Can you see my screen?
Yes.
Thank you. Good morning, everyone. Today, I talk about fault chain detection in the biological systems. This paper is a review of articles that deals with the mathematical description of biological systems under the approach of change in the relationship between external stimulus and system response. The biological systems, sensory systems in cells include signaling pathways and the bacterial chemiotaxis systems that give motion toward attracts and sensory systems in animals include vision, hearing, and smell. This review focuses on a property that many sensory systems at different scales and across organisms share cold chain detection, FCD. FCD. In these figures, we can see that the quotient between input and time is the same. For example, in 25 here and 5, the result is 5. 25% with 5, the result is 5. In the similar way, between 5 and 1, the quotient is the same. In this case, if one system satisfies this relationship, we can say that that system is FCD, fault chain detection. And the output of that system's output versus time is the same response curve, independent of the absolute change in that input. Only depends the relationship between inputs. The output is the same when the relative time input is equal. Figure 1b. Figure 1b, when multiplying scale the input by a scale factor, in this case, this signal and this signal, the difference is by scale factor lambda. The output remains equal. In this case, in the amortiguate dynamic. The first dynamical measurement that explicitly demonstrate FCD in dynamics were presented by the Shimizu laboratory using E. coli chemotaxis by means of the microfluidic setup. And in this table shows some biological systems that are FCD dynamic. For example, bacterial chemotaxis with E. coli and nematode chemotaxis C. elegans, Andre and Gio, that works satisfy the FCD dynamic, and the experiment and the computer simulation are satisfy dynamic very well. Also, mammalian cells, Anita, Goose, mammalian cells mentioned here fulfill dynamic. I don't know in the details, but it's very interesting to see and read about it. Finally, there are two types FCD dynamics, two types. In these figures, I show A and B, the incoherent dynamic and nonlinear dynamics. Twice are described for ordinary differential equation, and in this case, the nonlinear approach, the nonlinear differential equation. An alternative description can be realized with the logarithmic approach, but the mathematical details are difficult from for the short presentation, but it is very interesting for study and research. Thank you very much.
Hello everyone. You can see my screen?
Yes.
Okay. I continue with the topic of the last week. I find this paper, it's very interesting for it's a review, but this paper describes the many alternatives for construct a different microfluidics devices with the biomaterial compatibility. Okay. For example, in this figure one, describe for how to construct and surface with a biomaterial and the biomaterial into the microfluidics device. Okay. For in the figure two, to describe and different strategy for make a different molecules and reaction for with cells. Okay, this find or describe that behavior for cells and the different states in the organs in a chip. Okay. In the table one, and describe a different strategy for construct with synthetic or natural biomaterial. And next, the example for how to construct the cells into microfluidics and reaction for the flows in the chambers, and reaction for co-culture or reaction with other type cells. In this case, real two biomaterial compatibility into the one micro devices. Okay, and the two types cells, the behavior cells, we need look at the similar and the most in vivo on in vitro, right? These the other examples for how to construct different micro devices and models and surface, and to when construct the different components, for example, agarose and collagen, rabin, proteins on extracellular components. Okay. The next, we can see how cells in behavior for rates, for example, on shear stress or different stimulates with in the experimental system. Okay, or others proofs in the system for 3D printer is other alternative for combined with microfluidic technology and 3D printer, for example, in the other cases in the cells, cancer cells, and construct in the 3D system for analyze the behavior cells. It's the other example, but how the cells in the Intel chip, what's the behavior of the it's environmental cells reaction for in chip with contain and bioprinting. Okay, that is other strategy for construct it's the other stimulate cells. Okay, and we can see that other biocomplemental reaction, and in this part we can see how cells in the how in the integrated and in the different bioprinting or microfluidic reaction. Okay, that is next week we continue this paper. It's along for how the strategy for tools on and I can see. Okay, that is all for this week. Thanks.
Okay, I'm going to share my screen. Can anybody see it?
Yes, we see.
Cool. Good morning, everyone. Today I'm going to talk about the blood cell image segmentation using deep learning semantic segmentation. In this paper, deep learning semantic segmentation cutting-edge technology is applied for segmentation red and white blood cell in blood smear images. A complete blood count is usually performed manually by specialists, and he may feel extreme fatigue as they have to count a large number of blood cells in blood smear images. Therefore, a computer-aided system is a necessary demand for supporting blood cell count. In this study, a convolutional neural network was used called SegNet to efficiently segment both white and red blood cells. SegNet architecture classifies all pixels in an image. The result is an image that is segmented by class. Now, we are going to talk about the proposed methodology. Regarding data set processing, peripheral broad smear images, an image manipulation program were used to label red and white blood cells for the purpose of segmentation. A part in each blood smear image is copied for creating the corresponding ground truth image. In this case, for example, Figure 1 provides information about the peripheral blood cell image and its ground truth in the data sets. About SegNet architecture, it is shown in Figure 2, utilize a pair of encoder and decoder to create feature maps for segmentation. Encoder architecture stacks convolutional layer, batch normalization, and using ReLU, rectifier layer unit for activation function. After that, max pooling is used to reduce the feature maps size. It's necessary for the output image have the same size as the input image. Therefore, the decoder stage include upsampling. Each decoder has correspond with an encoder. The last decoder is connected to a softmax classifier to classify each pixel in an image. On the other hand, here in Figure 3, the proposed message, the first step is labeling all of red and white blood cells in input image with different colors. This step utilizes a SegNet to label each pixel with its corresponding class, and then all leukocytes and all erythrocytes are separated into individual images. Regarding experiment result and discussion, this paper focuses on the segmentation of three classes in an image. For visualization, a pixel label image is overlaid on top of an original image, as you see here in Figure 4. Red blood cells are labeled as the blue color, white blood cells are annotated as the green color, and the background of the image is yellow. The areas with no color overlay, which indicated by red arrow, don't have pixel label. These areas are not used during training. At the same time, as shown in Figure 5, image have more red blood cells background pixels than white blood cell pixels. This imbalance of the number of pixels can affect to the learning model on the training phase. That's why class weighting is utilized to balance classes. With this in mind, Table 1 shows the semantic segmentation metrics to evaluate the quality of the model on testing data sets. In Figure 6, a confusion matrix of the proposed message on the testing data set is shown. True positive for the white blood cells, red blood cells, and background class correspond to the diagonal values. And finally, Figure 7 shows in A the original image, in B the ground truth image, and C the predicted result. In 8, in addition, in 8 is separated in two images. One of these includes all the red of blood cells, and another image contains white blood cells, whereas and D and E show the final results of the cementation phase. So that's all. Thank you for your attention.
Hi everyone, I'm going to be presenting this paper. And in this work, the authors develop a shape and color switchable block copolymer particle in response to a light signal. Their strategy starts from the design of photocleavable surfactants, which undergo a remarkable change in amphiphilicity and interfacial activity under light irradiation. It was hypothesized that the preferential interaction between the copolymer particle and surfactants can be tailored by light irradiation to transform the interfacial properties and therefore the shape of the particles using these surfactants. Under light irradiation, the photocleavable group on the surfactants was cleaved, and with the overall conversion to a carboxylic acid-terminated surfactant reaching approximately 92% in 2 hours. So first, having characterized the photocleavage of the surfactants, they implemented them to stabilize particles and facilitate a light-triggered transformation. So first, onion-shaped particles were prepared with NC-tab surfactants. The block copolymer particles were exposed to different times of UV light. And before irradiation, onion-like spherical block copolymer particles with photocleavable groups at the outermost layer were obtained. Subsequent light irradiation led to a transition of the overall shape as well as the inner morphology of the particles. And, well, for example, after irradiation for half an hour, the particles show a mixture of axially and radially stacked lamellar morphologies, forming tulip bulb shapes. Prolonged irradiation resulted in the generation of strip spherical particles after 1 hour. And as the time of the light exposure increased to 2 hours, inverse tulip bulb-like particles and spherical particles were produced. As seen in this figure, the ratio of conversion determined the particle shape and morphology. So, to demonstrate the potential of the wavelength-dependent shape-switchable behavior of these particles, they developed a model system to prepare particles with a mixture of 2 light-active surfactants having different activation wavelengths. Similar to the NC-tab surfactants used before, cleavage of the C-tab also leads to generation of carboxylic acid groups. Accordingly, the use of C-tab surfactants in the preparation of the particles produced a similar light-induced shape transformation, but with the use of 420 nanometer light to trigger this transformation. They monitor the wavelength trigger shape change of the particles that contain the mixture of NC-tab and CC-tab surfactants at a molar ratio of 1:3. As expected, the particles were initially spherical with the photocleavable group on the outer layer. Irradiation at 420 nanometers led to the transformation of the particle shape from spheres to onion-like morphology to spheroids with stacked lamellar morphology. During irradiation at 420 nanometers, CC-tab molecules were selectively converted to CC-tab with carboxylic acid groups. NC-tab surfactants were stable under 420 nanometer light irradiation, so there was a second step, a second irradiation at 244 nanometers that decreased the ratio of NC-tab, and an inverse onion-like spherical particles were obtained. Therefore, the shape transition of the particle was achieved first by irradiation at 420 nanometers, followed by another step, another shape transition at 244 nanometers, highlighting the excellent wavelength selectivity of the shape transformation of these particles. Also, the light cleavable group of the CC tab is strongly emissive, imparting tunable optical properties to the particles and allowing monitoring of the light trigger shape transformation by a color change. The spherical particles show a strong blue emission peak at 440 nanometers, and after irradiation for 3 hours, the particle deformed to a striped spheroid, while the fluorescence decreased by 74%. After prolonged irradiation of about 6 hours, the suspension of spherical particles with reverse onion morphology became nearly colorless, and the fluorescence intensity decreased by 92%. And finally, to demonstrate the potential of the light trigger shape control of black copolymer particles in patterning features with microscale resolution, they fabricated hydrogel films containing these particles and irradiated them through a photomask and examined the local shape and fluorescence characteristic in the exposed and the unexposed regions. And given the good dispersion of particles within the hydrogel matrix, each particle can act as a photographic pixel. So a square like this, a photomask with 100 micrometers features was placed on top of the hydrogel film and was irradiated. And spherical particles were observed in the light unexposed region. In contrast, ellipsoids were observed in the light exposed region. Consistent with the previous results, it showed a reduced fluorescence upon the conversion of the CC tab, and the fluorescence intensity decreased only in the light exposed area, as seen by the dark squares in the photos. And this would be all for today. Thank you very much.
Okay, thank you. You have a question?